So today I will be speaking about explainable AI in neuroimaging. So the first, the outline of my talk is as follows. So first we will do a short explainable AI tour, recalling some main concepts and ideas. Then we will focus on neuroimaging. And I will, in particular, show you some examples on which we are working. So the idea is basically to learn by example how this framework of explainable AI can be exploited in neuroimaging. So this is the big picture in neuroimaging. In neuroimaging, we have many kind of data which are very diverse and are diverse in terms of modality. As you can see, we can have functional MRI, fractional MRI, other kind of data, for instance, microscopy, up to genetic data or physiological signals such as EEG, which is not presented here, or even clinical scores, so spanning different dimensionalities as well, from 1D to ND. And also we can have descriptors of higher level, such as network parameters, when talking about the brain connectivity, for instance, as represented here. So the idea is to jointly process all this data to make sense of the brain as a tool, investigating and modeling his structure and function. Explainable AI is about understanding the why and the how of the outcomes of data modeling techniques and algorithms. And of course, it is essential in domains that are sensitive, such as the biomedical field. What explainable AI tries to do is to bridge human and machine intelligence, enhancing their communication. A lot of work is been doing in this context, but what we need, in our opinion, is a pragmatic approach, saying that it allows us to define what we want to do exactly with explainability and interpretability, and how to assess it. This concerns the models of different levels, so of complexity, from very low complexity models, such as linear models, as the one displayed on the left, to high complex models. And of course, talking about high complex models, we immediately think of deep networks. So all this kind of model is concerned by this explainability issue that we are now trying to illustrate through examples. The basic pipeline that has to be faced when we talk about explainability in machine learning and deep learning is as follows. So first, we have to define the problem that we want to solve. This problem can be classification, can be regression, or also can be association studies. So this problem uses different kinds of data, as we have already described before. And depending on the data and on the problem, we have to choose a model to be able to answer our scientific question. This model can be linear, multivariate, can be statistical machine learning, can also be neural networks. And so the next step is to define the performance assessment method that will be used to assess actually how this model is working in our data to solve our problem. The parameters for performance assessment, of course, depend on the model. So for classification, we will be talking about accuracy, sensitivity, specificity, and so on. While if we are facing a regression problem, we most probably will be interested in mean absolute errors. While in association studies, we will deal with correlation coefficients and their significance. So once we have the performance, we want to derive some explanations that allows us to understand how the model got to this performance, and how this could be explained to humans, for instance. So in general, there are different methods that can be used. We are specifically focusing on those methods that allow feature probing. Feature probing means either feature ranking or feature visualization through the use of activation maps, which are by far the most exploited in the neuroimaging field. So once we have done this, we still need a step because we need also a validation of the explainability method that we have used and the explanations that we have derived to get an idea of the trust that we can have on these results. Validation is relatively low, poorly investigated so far, but it is really important because it is the step that will actually enable to reach trustworthiness in the solutions that we have devised. This validation could be performed in different ways. There are some approaches that have been proposed. And among these is the evaluation of the concordance with knowledge, the association analysis, or the assessment of properties such as stability and robustness of the descriptors that we use as explanations. So the core question that we are trying to answer is how to make sense of what we observe and whether we can trust these results. This solution is quite tough and also implies the definition of concepts, attributes, and their properties. For instance, concepts means we use terms like interpretability, explainability, and the rich ensemble of other terms that are appearing and that need to be defined. So there is still a lack of agreement in the use and in the choice of these terms. Then we have to define some attributes, which allows us to qualify the XAI model that we are using, and to define explanations and the attributes of the explanation that allows to decide whether an explanation is good or bad and how to assess it. So pragmatically, we need a clear and consistent syntax, fixing the terminology, proxies for deriving explanations, relying on the definitions and measures that we have chosen to use, and methods for assessing the quality of the descriptors that we derive, so for validation. Okay. In our terminology, we mostly rely on two terms, which are interpretability and explainability, that are summarized here. So interpretability to us refers to the ability to explain or to present in understandable terms to a human the outcomes of an algorithm. So it is related to the concept of causability, while explainability is associated with the decoding of the internal rules and mechanisms that are inside the machine learning system. So interpretability is related to causability and does not axiomatically entail explainability, and vice versa. An explanation is a presentation of some aspects of the reasoning, functioning, or behavior of a machine learning model in human understandable terms. There are very many types of explanations that can be used. We rely on feature probing methods, which provide explanations which belong to two main categories, that is feature ranking and feature visualization. I will provide some examples of this in what follows. Regarding validation, as I was mentioning, there are many attributes that could be probed as well, but we will focus in this case on stability. And I will provide you an example of this at the end of the course. Just to remind some definitions that you encounter every time you look at any paper in XAI, there is a taxonomy that is quite agreed, distinguishing between ante-hoc and post-hoc models, depending on the fact that the model itself is intrinsically
understandable and so interpretable, such as a linear model or a random forest, or post hoc, meaning that you have to apply some interpretability and explainability models to the outcomes of the algorithm to make sense of what you observe. Along the same line, without going into the details, we can distinguish between local and global models, or model agnostic or model specifics. And we have a lot of different ways of probing the features, among which, as I was mentioning, there is the visualization through heat maps. Just to mention the relation between explainability and performance of the algorithm, in this nice paper, which is quite recent, they show this summary in a nice graph, showing that in general, highly explainable models have lower performance and vice versa. So models that are able to provide very good performance, such as deep networks, are most difficult to understand and to explain. Again, this is a summary, always in this paper, of the main kind of XAI methods that are used in the healthcare and medicine field. And you can see that there is quite of attention in this respect that is increasing, and that mostly regards brain and breast imaging. This is an increase of the number of papers published per year in the medical image analysis field regarding XAI. And as you can see, and as I was mentioning before, the large majority of these works concern visual explanations. Because in the biomedical field, the final user, the final user is a human, and so you have to be able to convince human that explainable AI is useful and AI can actually help in solving everyday's problem and to advance research. So, looking at the biomedical imaging, there is some domain-specific knowledge that has to be taken into account and cannot be overlooked. In particular, besides providing explanations that are comprehensible by human, we have to demonstrate that the explanations that we get are based on relevant neurobiological signals and not on artifacts or confounds. To get to this point, normally validation is performed, and mainly and historically via post-hoc associations. So looking at the association between the facts that we find in these in our algorithm, for instance, the feature ranking that we get, and the role that each of these features is known to have in the particular problem that we are targeting. More recently, other methods are emerging, such as feature visualization through heat maps, and one of the most used tools is layer-wise relevance propagation that I will also use in my following example. In the medical field, there are other specificities that have to be taken into account and that are not common, for instance, for computer vision problems that you can have to face in another context. The first is the presence of confounds. Confounds are parameters that affect the outcomes of your algorithm, that could affect it, but that are not of interest for the problem that you are managing, in the sense that they are known to have an impact, and the results that we want have to be independent on these factors. The most used and known confounds are, for instance, age, gender, level of education, and then, depending on the specific case we are looking at, possibly others. So confounds are not explicitly treated in and processed in, for instance, in deep learning. So we have to be careful when we pretend that the results that we obtain are free from the impact of these variables. Then the data are multimodal, multidimensional, they are affected by missing data, for instance, they are noisy. And also sometimes there are privacy and security issues that impede sharing of data, and so to build models that are enough generalizable unless using data from public repositories or private databases. Okay, this okay, I can off to, sorry. These are the three case studies, showcases that I want to illustrate in the remaining time. That regard respectively imaging genetics, neuroimaging, and validation. These are complementary in a sense because in the first example, we will investigate association in an imaging genetic framework using a very simple linear model. So we are in the left end of the first slide that we saw, while in the neuroimaging example, we will be dealing with the classification of three-dimensional neuroimaging data, so we will be using 3D CNN. In the last part of this lesson, I will illustrate you a small study that we did to start facing the problem of validation, focusing on stability. So let's start from imaging genetics. Imaging genetics is a field where different kinds of data are gathered together, and in particular genetic data and imaging data. So from the imaging side, you can consider all scales of description for the brain structure and function. So you can start from microstructural descriptors, such as those that you can obtain from diffusion MRI, and functional signals, such as those that you can measure, for instance, using bold fMRI. And then you can go from this first level to the level of biophysical modeling, or regarding the structure, or task or resting state functional MRI localization. And then up again to the level of brain connectivity, structural and functional respectively, and from connectivity to networks. So modeling these adjacency matrices representing connectivity with graphs, and extracting graphs from graphs some measures that are eloquent with respect of the pathology, for instance, that we are considering. So all this data can be put in relation with other kind of information that is extracted from the genetic profile of the subjects, and putting this together allows to identify and to characterize the link that there could be between these two microscopic and microscopic to macroscopic domains. So in this case, the pipeline that we saw in the beginning instantiate in this way. We have to face an association study linking genotype with imaging phenotypes. We use a linear model that we will see will be a PLS. We want to study association, so basically we will face the calculation of correlations and explain variance. And the explanations that we will derive from this model are substantially the model weights, because we are in the case of very simple of a linear model. Then to assess the validation of the conclusions that we derive, we could use specific tools to this condition, which are, for instance, the assessments of the significance of the model, for instance, by a permutation test, or also the concordance of the results that we get with prior knowledge. So a bit more in detail, we could consider any of these imaging endophenotypes that span across the different imaging modalities that I have described so far, at the imaging level, at the connectivity level, considering microstructure, and also looking at either voxel or region-based levels. In the particular study that I will illustrate in this case, we only use structural functional MRI, and so morphometric information. From the genetic side, again, there are many scales at which you can look at the information that you can derive, ranging from the SNPs, so the single nucleotide polymorphisms, which reflect differences in single nucleotides, or you can aggregate these SNPs at different levels, from the gene level, such as using the sequence kernel association test or SCAT values that are aggregated then at the gene level, or two polygenic risk scores, which combine the effect of multiple independent genetic variants in a unique descriptor for a subject, representing the risk, for instance, of developing a given pathology. So we investigated the genetic mechanisms underlying brain atrophy in the Alzheimer's disease in the continuum relying on a data-driven PLS multivariate approach that I will explain in a few minutes. The objectives was uncovering the association between imaging and genetics determinants, considering a cohort composed of controls, healthy controls in patients, so Alzheimer's disease patients, and analyze the common latent representation, focusing on the components showing a clear separation between the classes. So what we did, we considered from the imaging side structural MRI, which allows to quantify global and local atrophic changes in the gray matter during early and late stage of Alzheimer's disease, and which has been shown already to be biomarkers for this kind of disease. So we chose some features that we knew a priori were really eloquent with respect to the disease that we were considering. On the genetic side, in this study, we use polygenic risk scores, which again have been shown to be associated with clinical diagnosis and disease progression. And also some previous studies could show the link with some imaging biomarkers. So we used a very simple model, which is the partial least squares. That is a multivariate method, which projects the data at input, which are the imaging and the genetic descriptors of our subjects, into a common latent space in which the covariance of the components is maximized. So what we have to do with the PLS is the estimation of these weights that project the data from the original native space to the latent space, and which act actually as the explanations that we are looking for. Okay, this is just to say that if you do an analysis of ADNI, you better rely on the ADNI database, which is an initiative started in 2004 and collects many images and other kind of data for Alzheimer's disease patient, healthy control, and also MCI, so mild cognitive impairment affected people, which is a kind of intermediate state between healthy and disease status. There are different imaging modalities and also genetic information. You can also use the biobank, but biobank only contains data of healthy subjects, so it is not immediately usable for studying Alzheimer's. So we consider 800 more or less subjects, and we performed this experiment using the PLS. For the genetic features, we use 14 PRS scores, And for the imaging features, we use 42 region-based morphometric imaging derived phenotypes, which represented subcortical volumes and cortical thickness derived from the structural T1 weighted MRI images. Okay, so again, from the two matrices of the IDPs from imaging and polygenic risk scores from genetics, we derive these weights and go to the latent space. In this latent space, the results look like this. So we have a set of weights relating the input features, so the cortical thickness of the different brain regions as are illustrated here, and the weights associated to the PRS scores. Here we identified two significant components in the latent space that were responsible for the 54 and 18% of the data variability respectively. And in this way, you can see which are the features that mostly contribute to the projection to the latent space, because the weight is higher, and also how these weights are correlated in a wide sense to the weights of the other component. For instance, here we can see in the first PLS component, there is an anti-correlation between the features measured in a given set of cortical and subcortical regions, illustrated here in green, and one or more of the less specific PRS. This is called less specific because depending on the way these polygenics risk scores are extracted, they can be more or less representative of a given disease. So this is the less restrictive condition. So the PRS is less specific. In the second component as well, we could observe an anti-correlation between the features of two specific regions, and also considering other regions as illustrated here. So the red means positive, the blue means negative. Of course, this is only an indication of the fact that there is a correlation or anti-correlation among the features in the latent space. It doesn't mean that one increases and the other decreases and so on. We can only say that if it is well known that one component, one feature, for instance, the cortical thickness decreases because of the disease, and we observe an anti-correlation, it means that in the genetic counterpart, there is an increase in the corresponding score. Okay. Also, you can interpret the results in the latent space looking at the projection of the data that you used in the latent space. For instance, here we used we can observe in the first and second component of the discovery set meaning the set that we have been using for training, consisting only of Alzheimer's disease patient and healthy control, a clear separation of the two categories of subjects, indicating that we were able actually in this space, in this latent space, to separate the two populations. On the contrary, looking at the testing set that was consisting of MCI patients, so patients in between the two categories, this distinction could not be observed. Another thing that you can observe in the latent space is the statistical difference group-wise in the components. So in the phenotype projection, so imaging features, significant differences could be observed on both the PLS component, as you can see here, also in the case of the testing set, while regarding the genotype projection, this could not be observed, though a trend towards significance was observed. So This example was to illustrate the fact that interpretability issue also concerns very simple models, such as the PLS, which is linear. This model is anti-hoc because the model is inherently explicable and allow to identify the features leading the correlation between the two variables that you are considering, so imaging and genetics, highlighting those having a higher impact in the latent space derivation. Okay, be careful that to get easily to this conclusion, you also have to perform a standardization of the data that you use. Otherwise, the weight that you derive are not immediately interpretable as actually relevance in the projection and in the process. In this case, the post-hoc validation was assessed by the consistency with prior knowledge and, as I was mentioning, through to the assessment of the associations that we could observe with known biomarkers of the disease. The fact that you perform validation is important because if you can show that some of your findings are in line with the knowledge, with the prior knowledge about the disease, then you can trust also those findings that do not still have a counterpart in the literature or in the prior knowledge, because they point toward, for instance, new biomarkers. So this phase of validation is really important, not only to trust the results that you have as a confirmation, in a sense, of something that is already known or a generalization of that, but also to discover new paths to follow and to define new biomarkers for the disease, for instance. Good, so I am a bit late, so I will be a bit faster in the second part. In the second part, we look at interpretable deep learning as a means for decrypting disease signatures in multiple sclerosis. In this case, the problem that we have at hand is the classification of different phenotypes of a disease, which is multiple sclerosis. The data that we use is three-dimensional descriptors, different MRI modalities I will describe in a while. The performance assessment is the classical performance assessment in classification, and the explanations that we derived are the heat maps from the LRP method. Then the important part, I think the interesting part here, is to see how we performed the validation. So divided in 3 steps that I will illustrate in the following. So just to explain a bit about the problem, we have 2 stages of disease that we call PPMS and RRMS. The PPMS is the worst one because it means that the subject is progressive in the disease, and so it's getting worse with time, while the others are stable. So we wanted to split these 2 categories because the earlier we can identify a trace of progressiveness, the better it is for the patient. So we used different descriptors from the images, all derived from diffusion MRI. In particular, we used some descriptors that we call RTAP, RTOP, RTPP, PA and NG, that represent the local diffusivity of the tissue, of the brain tissue. So in the brain tissue, you can identify some diffusivity profile in each voxel in particular, and this diffusivity can be characterized by these parameters that you can estimate through specific models. We used also other features that were derived from always the diffusion MRI, but are rotation invariant, and give some maps that are slightly different from the one before. These are particularly interesting because being rotation invariant provide the disease tissue signatures which are invariant actually, so more robust as descriptors with respect to the other ones. We also use some other typical parameters that you can extract from a simple model for diffusion, which is the diffusion tensor imaging, which are FA and MD. As you can see, all these maps show a different contrast between the white and gray matter, and if you look closely, a different description of the microstructure of the different tissues. So each one of these is a three-dimensional volume, and we trained different 3D CNNs using all these data as a single channels to this 3D CNN. So we had many 3D CNNs, one for each index with a particular architecture, and we applied then the layer-wise relevance propagation algorithm to the outcome of this network. This is the performance in terms of accuracy, precision, and so on of the different networks, and relying on the different indices, showing that actually regarding accuracy, the structural T1 is still the one providing the best performance. So the first step toward validation was to be sure that the results that we could get were not led by the confounds. Actually, as I was mentioning before, it is not usual in deep learning to perform the confounding before applying the algorithms of deep learning, the CNN. But in the end, you have to be sure that the results that you get are led by the parameters that you are targeting and not by these confounds. So the method that we used was inspired by this paper of Ding et al. Of 2020 and consisted in contrasting two logistic classification models through the likelihood ratio test in order to assess the impact of the main confounds, so age, gender, and EDSS, which is a disease-related score. The two models comprehended either only the confounds or the confounds plus the outcomes of the algorithm, and the statistical significance of the likelihood ratio test could reveal that the results were not dominated by these confounds. So we could be confident that actually the features that we were targeting, in this case the T1, was the T1, the DTI entry and other model derived Shor indices were the ones leading the classification. Then the validation steps 2 was to apply the layer-wise relevance propagation that you have already analyzed in the previous lecture, so I will not go into the details of this method. But basically, we, for each subject in the cohort, we derived the maps representing the LRP maps for the two conditions, PPMS and RRMS. So the first one speaks about the features that mostly witness in favor of being RRMS, and the second kind of maps talk about what speaks for PPMS in a given subject. So this is an example of the maps that we got. We have two different columns in each figure. As you can see, the two figures are for different microstructural indices, PA, RIF1 and RIF2 in the first and the others in the second. And these maps, for instance, on the column on the left, on the right of the first figure, are the relevance maps speaking in favor of the PPMS for the subjects. These are maps averaged across all the subjects that were true positive for this category. So we can see that there is a different kind of pattern for the two for the different indices. And another thing that it is important is that we analyze these LRP maps also considering the maps for the non-target class. So what does it mean? The LRP takes the value of the decision function at the output of the network and projects it back. So if I take a subject, which is a true positive for the class PPMS, this value would be, of course, larger than 0.5. Propagating back this value, I will get this mask. But also there is the 1 minus this value that I can propagate back and get this other mask, which is the relevance for the non-target class. And showing this, we can have an idea of the differential weight of the different brain regions in the two conditions. Actually, as we know, higher contrast reflects higher levels, higher relevance, and actually, for the non-target class, for the reasons that I just mentioned, the mean of the activation map is lower than for the target class. We wanted to further exploit this information by looking at the difference in these relevance values in the different regions, ROI-wise. And so we wanted to identify the regions playing a prominent role in the classification between the two MS phenotypes, which were the regions of higher LRP across groups. So the point is that we wanted to interpret the LRP maps not based on the absolute values of the relevance, but on the differences and overlaps between the violin plots of the considered ROIs in the two classes of patients. We then analyzed these LRP maps, and we got this kind of plot representing on the horizontal axis for each index the different regions, and on the vertical axis the average relevance value. In all this, we could identify two regions that were consistent across the different plots, and so across the different indices, that were the temporal pole and the parahippocampal gyrus. So in this case, you can see the plots, the violin plot is shows a higher relevance. And also, if we look at this other set of plots, which represent the differential gain, so the difference between the mean value of the relevance in the regions in the two conditions, so target class, non-target class, we can see a high distance. So what we could conclude is that actually there are some regions that are leading in this distinction between the two classes of subjects, and that the relevance of the regions is different depending on the fact that the class is the target class or not, and this difference is maximized in specific regions which are eloquent with respect to the disease. So this witness is in favor of the soundness of the observation that we did. Then another step for assessment of the soundness of what we did, so validation, was the association analysis. We know that some of these microstructural indices are eloquent with respect to the disease. So in particular that the RTPP is statistically different in the two categories of disease, in the two phenotypes of disease. And so in order to strengthen the significance of our result, we calculated the statistical significance of the correlation between the mean value of the significance map for this parameter in the hippocampus and the RTPP mean value in the hippocampus, which actually was already shown to be eloquent for the disease. Since this association resulted to be significant, then we could conclude that actually the results that we got were sound. So just to conclude, can we trust the explanations? Besides these validation steps that can be performed at the end of the algorithm, a problem remains about the fact that we have the doubt that if we change the data set, or if we change the network, or if we change the hyperparameter of the network, we get different results. And so in fact, we cannot really trust what we are looking at. So a first step in this direction was taken always regarding the classification of healthy versus Alzheimer's disease patients, and comparing the descriptors, the explanations that we could find as responsible of the features responsible for the classification, using the SHAP values as descriptors, and using 5 different architectures for the classifier. So based on this, we defined a new proxy that we call normalized movement rate, that try to assess whether the ranking of the features that we get is or not congruent, consistent across the different classification algorithms. This is the architecture pipeline that we used. So 25 IDPs, always taken by the structural MRI, 5 models, a 5-fold cross-validation architecture, calculation of the SHAP values for each model, and ranking of the features. Then we focused on the first 10 features, and what we did is something quite simple. So taking the first 10 important features as a reference, so we only considered the first 10, we progressively removed all the first one, so the most important one, and rerun to SHAP on the, rerun the algorithm and rerun the SHAP to see whether the order or the feature would change or not. Then we counted the number of changes in the position of the features, and we normalized it in a given way, as illustrated here, for each classifier. So basically, the less changes, the best. The lower number of changes, the best. In this particular example, this number is 0.36. So this is what we could get across the different classifiers. The SVC had the lowest value for this NMR index, while the others had values which are higher for this index, meaning that removing one feature at a time would make a larger number of switches, of permutations within the features that one could derive and rank. Among the different models, the decision tree was the one that performed worst. Another thing that came out that is interesting, and you have to take into account if you work in the biomedical field, is that this switching in the features strongly depends on the collinearity of the input features. And to have an idea of this, we repeated all the procedure by making a PCA before the whole process. So if you do a PCA, this switching of features in repeated runs of the algorithm decreases significantly in all cases except the decision tree. And in particular, in this case, for the SVC, we didn't record any changes in the order of the ordering of the features in this case. So be careful not only to confounds, but also to the collinearity of the features. Then in the end, we wanted also to evaluate the agreement across the different classifiers. And we could find that the correlation among the ordered list from the different models were generally low, indicating that each model produced different rankings and scarce agreement between sharp results from different classifiers. And this improved a bit after applying the PCA, which increased the amount of the agreement across the models, as it was reasonable to expect based on the previous results. So, to conclude, in our view, XAI methods are, of course, increasingly used in neuroimaging and related field and are the enabling technology for AI solutions. But there is still work to be done for reaching an agreement in the terminology and taxonomy of the model. But even more importantly, validation is crucial, is still in its infancy, and calls for methods and proxies to evaluate features which are of prime importance, such as reliability, robustness, stability, fairness, and so on, but also the relation to uncertainty and to accuracy. And last but not least, causability. So there is no golden rule, and care must be taken in the interpretation of the outcomes also of XAI, keeping in mind that what we are measuring cannot be linked in a straightforward way to underlying neurological and neurophysiological mechanisms, and even strong associations can be misleading. So I want to conclude with this well-known example, which shows the correlation between the number of people who drowned by falling into a pool to the films where Nicolas Cage appeared in, and to cast some doubts on your mind. So thank you for your attention.